Hi, good afternoon, and thank you so much for the opportunity. My name is Michelle Martinez, and I am a professor in the Department of Biochemistry at UCC School of Medicine in Puerto Rico. And I am also president and co-founder of Dynamiko Pharmaceutics, a women's-owned startup with the mission of developing selective anti-cancer therapies. Now, I'm sure that in this audience, everybody knows someone who has been diagnosed with cancer and someone that has died from cancer. So I'm going to tell you about my friend Elsa, who passed this past March from triple negative breast cancer. Elsa was a young woman in her 30s. She was a recent mother, and she was one of the 2.3 million women diagnosed with breast cancer globally. Elsa was also part of the 460,000 women diagnosed with the triple negative subtype, which is a very aggressive subtype that has increased its chances of death. And this is because 70% of the women that have triple negative breast cancer, in those women, the current therapies do not work. And why is this? This is because current therapies are generalized. They cause impactful side effects, and they are also expensive. Some of these therapies required clinic visits or hospitalizations that decrease patient compliance. Their inefficacy also increases the chances of the cancer cells to spread to other parts of the body, which is commonly known as metastases, and this drastically decreases the chances of survival. Our solution are EPDs, or ergosterol peroxide derivatives, which in Dynamiko we are formulating so they can be taken home in the form of a pill. Our innovation is based on our discovery of a molecule found in this medicinal mushroom, Ganoderma lucidum, which we have been studying for the past 15 years. EPDs target a protein that is overexpressed in triple-negative breast cancer tumors. Our data shows that EPDs are efficacious and that they're safe. We get 60% reduction in tumor volume, and important to mention that 3 mice in this trial did not have any tumors at the end of the study. Furthermore, as you can see from your graph on the left, liver profiles show that there is no toxicity, and we recently published our safety data, which includes maximum tolerable doses of 500 mg per kg of body weight, and we did this both in males and females. Also, no cardiotoxic effects and no hepatotoxic effects as well. Very importantly, our data also shows that EPDs reduce the spread of cancer cells to 2 very important organs that commonly have metastases in triple-negative breast cancer patients. And these are the lungs and the livers. We get a reduction of 84% of metastases to the lung and a 73% reduction in metastases to the liver. Therefore, EPDs are selective therapeutic agents for this patient cohort. Our market is based on the number of patients diagnosed with breast cancer globally multiplied by the cost of treatment per year, and it comes out to be $230 billion. The serviceable addressable market is the 20% of those patients that are for the triple-negative breast cancer cohort, comes out to be $46 billion. And the serviceable obtainable market is for those that fail therapy or succumb to disease, and it comes out to be $23 billion. Our competition are both the chemotherapeutics and the biologics, and they both have low efficacy, primarily because they have multiple impactful side effects and they are not selective. In terms of the biologics, although 30% of the triple-negative breast cancer patients do respond to biologics, many of those new reports show toxicity, and also they are very expensive. In contrast, our EPDs are selective to a protein that is overexpressed in this cancer. And moreover, our compounds have the great potential to be effective in an oral formulation. We are an early drug discovery and development company focused on an asset-centric strategy, aiming to provide high-quality preclinical and clinical data to submit an IND to the FDA. Our team combines oncology and pharmacology expertise. I have more than 15 years experience in natural therapies in cancer models. My PhD is in nutritional genomics and animal science, and I have 2 postdocs in functional genomics and also in cancer biology. My co-founder, she's Fatima Rivas. She is an assistant professor in the Department of Chemistry at Louisiana State University in Baton Rouge. She has 20 plus years in research in natural product-based therapies, and she has a PhD in medicinal chemistry and postdocs in organic chemistry. She has a pharmacology background, and our multidisciplinary research expertise aligns very well with our mission in Dynamiko. We have achieved so far $750,000 in non-dilutive funds. We have 4 grants currently. We have 1 PCT patent that was published in 2020 and that it nationalized in the US, in Canada, and it was recently issued this year in Japan and in Europe. We have 3 publications for EPDs, many more in the medicinal mushroom space, and we have presented our research in many scientific conferences. We have been part of BioLeap, which is an innovation accelerator program for life sciences in Puerto Rico, and currently we are in the 2nd cohort of IRED, which is an NIH-sponsored commercialization program. We're currently fundraising for $500,000 to conduct additional preclinical studies that we need to conduct to have the necessary studies for the IND submission. Moreover, we want to purchase instrumentation that will allow us to conduct additional biophysical experiments that we need for target validation, such as an ITC or a monolith for microscale thermophoresis. Thank you so much for the opportunity and for supporting our efforts to developing life-saving therapies for cancer patients, so future women like Elsa don't have to suffer the same fate. Thank you.
Thank you, Dr. Questions? Yes.
What was the frequency of the dosage in your animal studies and for how long was that study?
The study was conducted, so the question was what was the frequency of the dosage in the mouse studies and for how long? It was given once a day via oral gavage for 11 weeks.
Thank you for repeating that question for us. Yes.
Is this a single isolated compound or is this a extract from ergot?
So the question was if it was a single compound. Yes, it is a single compound that we no longer extract from the mushroom so we can use it. We synthesize it. We have methods to do that.
Fully synthetic pathway. Any other questions? Yes.
Did you ever stop dosing to let the tumors grow back and then redose to make sure that there's no resistance being built?
So the question is if we stopped dosing to see if the tumors grew back. No, we haven't done this with EP derivatives, but we did that with the mushroom, and they do not regrow.
Excellent. Well, thank you so much,
Thank
Dr.
you so much.
Martinez. We really appreciate it.